It'd be a shame not to record the last one. Okay, well, um, anybody that wants any time to review any of this, you can, I don't make it public. I haven't made it public, but if you email me, I'll give you permission to look at these. And I tried to get Heather in, she's in today, but to realize that you're better off to have the slide set that goes with it. Now, speaking of that, there's 365 slides in this presentation, in this thing that we started in January. If anybody wants the whole set in PowerPoint form, which is with my notes, everything. I mean, the whole enchilada that I complete with, I don't think there's anything in there embarrassing in my notes, but I don't really care anymore. But I can make it available. Maybe I had decided where the best way to do it is put it up on Microsoft OneDrive and give you permission to put it up on Google OneDrive. But I would try to do that. I think if anybody wants it, you could email me about that. Now, Donna has a smaller version, the one that Linda Chafin made me cut it down to try to give this. Can you believe that I had to give 300 slides in one day? Give me a break. But anyway, I did what she told me. I packed 10 pounds in a 5-pound bag. All right. So Jackie's laughing, crying too. Jackie, have you finished that course for the certification?
No, actually, I'm taking the last class, which is basic botany. That'll be in November. So I'll have to give you a report on that.
Well, now you betcha. You know why? Because guess who gave the basic botany? What they did was you might be seeing some of my slides.
Oh, cool.
I hope they give me permission or give me credit, but I don't know who's giving it, but I don't envy anybody doing it, because the trouble with something like that, I don't want to go on too much, is that, first of all, if you cut out too much, you don't make the connections between the subsciences.
Yeah.
Hey, Bob.
Just like we're doing plant evolution, but before that, Heather, we did plant genetics, and before that, we did plant reproduction, and they all tie together. And so it's kind of like a puzzle. What's that there?
Excuse me, Bobby. This is Steve. I think you renamed me Donna.
Excuse me. I did. That's funny. All right. Steve. I always want to put Stevie Wonder because it's
you can do that. But I prefer to be called Diane.
Where's Donna? What happened to her? Oh my God, she's not here. Oh, there she is.
I'm here.
iPad. Wait a minute, you keep jumping her. What happened? A minute ago, you were further down on the list. Okay, here we go. All right, having these boo-boos. All right, Donna. All right, so, all right. Okay. All right, now let's get on to it here. We're gonna, yeah, Jackie, you'll have to tell me, and we won't get after them if they use my stuff. All right, now let me share the screen. That's the first thing I need to do. Okay. And I'm gonna do this annotate bit. Okay, this little gadget, this one. Now I'm gonna bring up, there's the PowerPoints. This is slide 323 out of 365. Now, this is not where we're starting. I'm going here for Heather's benefit mostly, because this is, it'll benefit you too a little bit, because last week, I'm not going to go to the show, to the big screen right now, but this is
Bobby,
where we start. I've
looked over the slides, so I'm fine to start anywhere.
Okay, but what we're doing, this is going to benefit them too, Heather.
Okay.
Because, so don't worry about it. Because this is where we started last week, having just finished plant reproduction. And we went through, most of the time we were talking about pollination mechanisms, and we got down to the pretty much right. Well, we that was mostly everything we talked about for the most part. Today we're gonna start on plant breeding systems, in and out breeding. Having some congestion here. And we're gonna finish up. And the last segment is propagule dispersal, which like how do you get seeds and spores dispersed? And we're going to go through propagule in a minute here. In fact, let's go ahead and wade through these. All right, propagule is a term that we brought up last time. We'll be using it again toward the end today. When we get the last half a dozen slides have to do with propagule dispersal. Any plant part, sexual or asexual, that becomes detached from the parent plant that can form a new plant. Examples of sexual propagules include seed, and that should be technically, as some of you know, myospore, M-E-I-O-S-P-R-E, a spore that comes out of meiosis, which is a sexual process, not a spore, not a mitospore that comes out of mitosis, but those had that. There's two kinds of spores. All right, now most of the presentation dealt with these three ways: water, wind, and animals. And we're going to just run through it. And actually, this was a neat thing too. Here at Willows, and I had forgotten all about this, but if I ever knew it. The willows have these. I mean, it's hard to think of these as flowers, but that's what they are. You got a bract, and you got stamens, and you got the pistil here that's stigma, style, and ovary. But here's a wind pollinated plant that's still hedging its bets with a little gland down here to attract pollinators. So willows aren't exclusively wind pollinated. All right, pretty smart plant for not having a brain. By the way, when you get to Florida, when you get to South Florida, that's all you got. The more north you go in willows, the more willows you get. In fact, Alaska's about half of them is willows, because up there, it gets cold. The pollinators are not too common. Now, okay, this is the oaks showing monoecious, where you have male and female separation you have stamens flowers and pistil flowers on separate branches, which is like one house. The tree is a house and the separate bedrooms, unlike willows that have two houses, house being the plant. And wind pollinated, okay, let's just, all right, this thing I showed you last time, there's a moonflower in Florida, South Florida primarily, but it flowers at night. Now, this is Ipomea, species in the morning glory family, sweet potato family. This was blooming out in my yard yesterday. Not this species was. This is a plant from Scriven County. And I didn't take this picture. One of our members took it and sent it to me. And I'm going to put it up on Discover Life pretty soon, but I just hadn't got around to it, given her permission. She's given me permission. That thing, in my whole life, I'd never seen it bloom. It's not that common. The original parent in my yard came from Fort Stewart. My friend Stella gave it to me, who died in February 2 years ago now. Year and a half. But I've got a piece of Stella in my yard, and this is it. She showed me the plant at Fort Stewart, which is about 5 miles as a crow flies from here to this plant. Bottom line, Ipomea macrorhiza. It's called the big root morning glory, and that's the root is really a rhizome. And so, but the funny thing is that I'm looking at the bird bath. And I tend not to look up because my neck hurts sometimes, especially in the morning when I was walking the dog. And I looked in the bird bath, and they had these corollas that were 4 or 5 inches long laying in the bird bath. And I look up, and there's just a bunch of these up there, and having just closed because it's daylight now. And but it was interesting that this thing in bloom I had never seen until till just recently, this weekend. Okay, but why am I bringing it up? Because this is a night pollinated or a moth or bat pollinated type of, and I do have bats in my trees here, so night pollinated species of plant. Okay, let's see, I think I was, yeah, that was it right there. Okay, let me get this thing out of the way. That's, you can't see it, but I can. All right, now, this was heather, the largest flower in the world. It's can be 3 feet, and it's nothing but a root parasite in Borneo, and it stinks to high heaven. And the flesh color on both of these is no accident. So Stapelia is a arid plant in the milkweed family, if you don't lump that family with the dog names, which is what's happened. Now, okay. All right, now this is where we're going to start out. We'll give examples of these 4 in a moment, and I'll start, when you start seeing the numbers, you'll know we're actually giving examples of the different kinds of mechanisms that prevent selfing. But first, we're going to talk about non-selfing up here actually, no, excuse me, we're going to talk about selfing. This prevents selfing. This promotes outcrossing or non-selfing, these mechanisms. But we're going to talk about inbreeding first, and it's selfing, union of gametes from a single individual versus outbreeding, transfer of gametes from one individual to another, genetically different individuals. Okay, the general advantage of outbreeding, we've already talked about this a little bit, is to promote an increase in phenotypic variability in the population. This generally enables plants to adapt to a wider range of environmental conditions and increases the likelihood for survival and evolutionary change. What we're saying is a mongrel or hybrid is better off in the wild than something that's purebred in terms of genetic variability. All right, we'll focus a little bit on inbreeding, and then we'll spend most of the rest of the time on outbreeding and wind up with propagule dispersal. All right, there's some 50-cent words here. Auto means same, and that's autogamy, bringing the gametes together within one flower. That is the extreme form of inbreeding and self-pollination, not cross-pollination. And Guyton, if you look up the word Guyton, and the rest of you have seen this book, and I'm going to show it to Heather, Dictionary to word roots and combining forms, is a little book that has the Latin and Greek and other words of which you could look up something like geit, G-E-I-T. In fact, it actually says G-E-I-T-O-N, Guyton, which means neighbors. So bringing together, so between flowers of the same individual is what technically, now that's a word I'll never use again probably, but it's a neat word. Autogamy or autogamy is a word that gets used quite a bit, but the genetic product of both of those is identical. All right, the good news about this kind of thing is the selective advantage is it ensures that the propagules get to where they belong. The pollen gets transferred to where it belongs, and it's not very dicey. It's not very chancy. And the disadvantage is that it reduces down to no genetic variability because you've basically got a clonal thing going on, but it's sexual. Clonal production is asexual. Now I'm going to show you something. Okay, well, let's see. Well, let me finish up with this. Allo referring to other kind of auto between both, involves both outcrossing and breeding in the same plant. Violets can do that. Some species of milkwort can do it where you have 2 flower types. In fact, when you're trying to key out milkworm species, they'll ask you about some of the flowers being cas. That means open, and cleis means closed like a fist. And violets are notorious for that. And the ones that are the weediest tend to be that way. And I think Viola sorori, is the common blue violet, does that quite a bit in my yard in Chicago. But so they're not very conspicuous because they don't open up and try to attract anything, just self-pollinate, don't really, then they just stay closed. No need to open. But that's good for a short-term situation. Now that brings me to, excuse me, brings me to this plant. This is a Clarkia. Clarkia is a Western genus of about 40 species in the Western United States. And it looks like, it's in the evening primrose family, and the four petals kind of tell you that. The closest thing that we'd have to that is Gaura, Gaura. And boy, look at those stigmatic crests. Those are just, pollen just sitting there almost as big as a flower, waiting for pollen to land on. Well, this thing, this is what happens when I typed in Clarkia and I typed in autogamy, and I got this. This is what I got. Let's see if I can. It's not going to let me bring it up, but it's not going to let me move it down. Thought I could do that. Let's see. Yeah, okay, I can. All right, I want to read this. I've never done this sort of thing, Heather, with them, but it's by performing these people out west performed an experiment, and they this suggests that different genes underlie each other in Clark population. Now, here's the thing: they have the ability to respond to the environment in several ways, and what happens is this plant. Let's see what. Geez, next page, I guess. Maybe. Well, I thought so. Oh, can't navigate this. I don't know what I'm doing. I guess this is why I didn't do it. But anyway, what it talks about, and I don't know why I can't get at it now because in terms of navigation with this, but this plant does autogamy. That's the reason when I Google Clarkia and autogamy, I got it. And what it does is pollinate earlier in the self-pollinate earlier in the early part of the season when there are no pollinators around, and later it shifts toward more of outcrossing or cross-pollination. And the earlier pollinated plant, the earlier younger plants in the spring tend to have a better photosynthetic rate than others too, so it grows faster. So this plant is adapted to the situation to self-pollinate when there are no pollinators around and switch more to cross-pollination when they are around. And it must work because there's 40 species of these things. And they're really tough to identify. So let me get out of that and go back to where we were. And the reason I mentioned that is it's a plant that has successfully done the best of both worlds. Because let's face it, people, any sex is better than no sex. And when it comes to survival, you got to do something, and then later on you can shuffle the genetic. Selfing works in a pinch, but in the long term, you have to have variability. Again, I've said this before twice, or at least, and now I'm going to say it for Heather's benefit. Asexual reproduction is a short-term asset and a long-term liability. And the plants that succeed both best are the ones that do both. And some of our worst pest plants are asexual, Kogan grass being one of them, and Johnson grass being another. All right, now plants in general and angiosperms in particular have evolved ways to prevent selfing, and that's what the next few slides are about. Remember back here, the number, there's 4. All right, well, that's there's number 1. If you're a dioecious, then you're going to definitely outcross. You have to go in the tropics where you have, if you walk around in a tropical forest, it's not like I see 10, 15 sweet gums and then maples and then this and that. You don't see replicates of individuals much. You see 2 or 3 individuals. And if you want to find the next individual, you may have to walk an acre or 200 feet and see another. And so if you're dioecious in the tropics, you really are playing a dicey game trying to find a mate unless you've got a good pollinator. This plant is a plant in our coastal plain flora, Florida rosemary or sandhill rosemary. It's not a mint. It kind of looks sort of like a mint in terms of the leaves being terete or roundish, but it's a pretty thing. But it grows in the most inhospitable conditions in sandhills around here, and it's dioecious like hollies. And then you have these plants are obviously brought together for the photograph. So, but I mean, what a pitiful flower. You don't have it. It's not very showy at all. You got these huge anthers on these stamens, and transferring it over to that stigma over there on the right, right there. So that would be cross-pollination on a dioecious plant. Now, this is still continuing, but what happens when you can get, well, actually, this is what they call a monoecious situation, at least here on this California plant. I don't know the situation about the eciousness, but over here on this member of the Chenopodium family, that you've got male and female, male down here and female flowers on the same main stalk, just different side branches. So that would be monoecious, like oaks, different branches of the same plant. But we'll come back to this. But you had pollen. I mean, what a pitiful flower. I mean, in terms of showiness, but it does the job. Pollen here will transfer to these stigmas. And if there's not some self-incompatibility problem genetically, pollen tube will grow down in there and fertilize the egg down in the ovary, the ovule of the ovary. Now, before we leave this slide, note another little thing that the plant has done. Not only, and this has to do with maturing at different times. That's what this whole slide's about. Having the pollen mature at earlier or later than on the same plant than the female. Now, okay, even if these were to mature at the same time, notice that the stamens down here with the pollen are below the stigmas. So that would, of course, this is pretty close from here to, that I can't tell, that might be a male. But either way, it's kind of like in a pine tree, you'll find that there's more pollen cones down lower than the seed cones, which would be more higher, and that's to promote outcrossing. So not only is this temporally separated in maturation times, but there is the idea that the pollen is down lower than not much, but than the pistils. So if the pollen was up here, pollen flowers, male staminate flowers are up top, it would just drop right in. So there's two mechanisms going on, one spatial and one temporal time to promote outcrossing. Now, this is a common plant, sort of, I call it one of the harbingers of the spring around here. The yellow jasmine, Carolina jasmine, state flower of South Carolina, which has strychnine in it and therefore toxic. But it has heterostyle, which is the same thing as diastyle, and that this flower here is like the one over on the left, except the stigmas are better shown on the actual photograph than the drawing. And then this one over here has a short style, and so that's another mechanism to promote outcrossing, diastyle. I I never try to remember which one of these is called the pin or thrum. I don't know why I even bother. What's wrong with long and short? Give me a break. You don't need those terms. Now, by the way, sometimes you have to do quite a bit of looking to find both of these in the same population, because it's not like 50-50. It's but on field trips, I used to give a prize or points or something to students that would find these first. But anyway, the fourth outbreeding example I'm talking about has to do with self-incompatibility, the ability of the plant to reject its own pollen. Now, it's genetically determined. Inability to fertilize occurs between gametes derived from one individual. It's the most common anti-selfing mechanism in the flowering plant world. Now, some plants actually reject pollen that has an S gene and matching an allele in the stigma cells. That means that it keeps the pollen tube from growing. And in other words, the stigmas of the flower have a chemical sense that detects pollen from the same flower and inhibits its germination or pollen tube growth. So, so that's where genetics get the manifestation of the genes gets manifested this way. So, all right. So now I'm going to give you a second to read that. Read these bullets. Maybe I'll just read it for you, because that way everybody is on the same page. I don't do this often, Heather, but I'm going to do it here. Any environment provides an arena for individuals within populations in which there are opportunities and hazards. Those members of the population that fit the environment thrive and leave more offspring than those that are less fit. Now, the example I have is population of herbs has a mixture of sweet and bitter individuals. And after herbivory in a pasture, say, over time, the more bitter herbs leave the most surviving offspring, because the sweet ones get eaten first. This is how natural selection takes place. In plants, they have no excretory system, so the alkaloids that are garbage build up. Can be caffeine, nicotine, those are alkaloids that build up, and the more they build up, the garbage builds up in these cells. The more bitter they become, the less palatable they become to grazers. So now, let's see. Okay. Now, how do these sources of genetic variation in population come about? How do you get variation? We're going to go over micro and macro mutations in the next few slides. Talk about gene flow, gene migration, and then this genetic recombination is something we dealt with back when we talked about sexual reproduction. You get genetic recombination at crossing over in meiosis and then reconstituting them in syngamy. But before we go further, we'll be actually talking about this gene flow idea when we get more, when we showed a slide or two on introgressive hybridization of oaks, which is why oaks can be so difficult to identify sometimes. But gene flow, gene migration is, let me see if I can find this. All right, think about this. I'm going to read this passage. Maintaining gene flow between two populations can lead to a combination of the two gene pools, reducing the genetic variation between the two groups. So what I'm getting at is gene flow can be negative in some respects, and that's what I'm getting to right now. For this reason that gene flow can strongly act against speciation. In other words, cutting down on species diversity by recombining the gene pools of the groups and thus repairing the developing differences in genetic variation that would lead to full speciation and creation of daughter species. Now, for example, if you have grass growing on both sides of a highway, pollen's likely to get from one side of the road to the other. And if this pollen is able to fertilize the plant on the other side where it ends up, it produces viable offspring. So the genes have flowed from one side of the road to the other. So two subpopulations in this area were separated by a barrier like a road. Then the alleles in the pollen have effectively been able to move from the genes, the alleles have been effectively able to move from one population on one side to the other. And just if that barrier between the two populations of grass was bigger, like an island or separation by water or something, they could speciate and become separate. But as a gene, what I'm saying is gene flowing back and forth could reduce them speciating, could keep them from separating into different, evolving into separate species. So gene flow can cut down on speciation. I remember when some people in Central Florida, Bach Towers area, were trying to grow these Dicerandra mints, and they were doing what botanical gardens do. They're trying to. They had a whole collection of them, and what was happening is that instead of these plants being spatially separated ecologically or geographically by distance, they were right there in the same building, in the same greenhouse. And guess what happened? They started interbreeding with one another and cutting down on variability. And in other words, the species started amalgamating into one or two things instead of three or four. And so when they brought these separate species together, they were messing with Mother Nature. And that was an eye-opener for some of those people. All right. These three sources provide the varied types of organisms needed before natural selection can produce the changes that we call evolution. Mutations, gene flow, and genetic recombination. The next few slides are these three bullets. Now, all right, I'm going to go to the animal world a little bit for this, but at the top is a plant example of a micromutation. We talked about last week, or actually 2 weeks ago, what a mutation was. But basically a change in the DNA, that's the simplest way we can say it. But now, these two T's, one of them is Fuller's, and I forget which one. I think the one on the right is Fuller's, but supposedly their distinction has to do with a micromutation. Now, a good example, the best example I know of a micromutation, let me minimize. Maybe you can't see that anyway. So, but let me minimize it for my benefit. All right. All right, we talked about insulin the other day as a protein that's made up of about 80-something amino acids. Well, I don't remember how many are in hemoglobin, but what's going on here is a difference in a person that has normal hemoglobin and a person that has sickle cell anemia type hemoglobin has to do with the substitution of only one amino acid in the chain, of which that is number 6. And glutamic acid or glutamine is the salt form of the name. Glutamine is the salt name of glutamic acid. They're the same thing. And the substitution of number 6 amino acid, glutamine, glutamic acid, by valine will bring on sickle cell anemia. It's something that simple. Now, this is looking at the proteins. Now, proteins are put together based on messenger RNA, and messenger RNA is a nucleic acid that gets this information from DNA. And this has to do with, if you go back to the DNA side of it, it has to do with the replacement of adenine for thymine in the codon. Now, I'm going to lose some of you here, but let me go. All right, this is something from a lab manual I used to have, and it's very good because it shows what a codon is. Last week we talked, or maybe 2 weeks ago, we talked about what the building blocks of proteins or amino acids, just like I showed you, but the building blocks of nucleic acids are DNA and RNA. The building blocks are nucleotides. And a nucleotide is made up of a nitrogenous base, adenine, thymine, guanine, or cytosine, and then phosphates and sugar groups. Well, a codon that's necessary, you need 3 nucleotides to make an amino acid. And so if this is the nucleus of the cell and you have adenine and cytosine, thymine, guanine repeating one another, but in an RNA, you have thymine being replaced by uracil. And so what happens is these 3 nitrogenous bases will code for a particular amino acid. There's only 20 biologically important amino acids. And so it's not hard to figure out the genetic code for them. But what we're saying is the normal hemoglobin would have the codon, it's not showing here, but it would have CAG, which is cytosine, adenine, and guanine. But what'll happen is the middle one, the adenine, gets replaced by thymine and will produce that amino acid. So replacement of one nitrogenous base in the nucleic acids will bring on this mutation. And we're really getting down to the molecular level of a mutation when we do that. I know I lost some of you there because we didn't go into that. We didn't go into protein synthesis, but now this is macro mutations. Polyploidy is a form of macro mutations. And plants do it often. Animals it just doesn't succeed with animals. And now here's an editorial point. With the gametes, most of you probably realize that this representative is really 4. These are really 4. So there's 4 of these and there's 4 of these because to get from diploid to haploid, you had to do meiosis. So what's going on here is going on 4 times. What's going on here from here on is also going on 4 times. But we're only looking at one of the products of this meiosis, which took the diploid down to the haploid condition. All right, so there's only one member of the homologous pair showing here and only one chromatid of it. The same is true here. Now, what happens is when these gametes meet, when running bear meets little white dove, we get a zygote. The zygote is sterile. Why? Because to do meiosis, which is the next step after syngamy, you have to have the next sexual step after syngamy. You have to have even numbers. That triploid can't undergo meiosis. So what happens, and this is where we'll see another example of this in a moment when wheat. But what happens is you get an abnormal form of cell division. Cell division consists of mitosis or meiosis followed by something we call cytoplasmic division, which is cytokinesis. So what happens is on the way to dividing, you get a reconstitution nucleus, and all the products of the separation get put back into the same nucleus instead of separated into 2 cells. So we have a failure of cell division, a failure of cytoplasmic division. So the doubling that takes place, this would normally go to 1 cell and then another. They all wind up in the same cell. And now guess what? We've got even numbers here and we can divide again. Okay, so instead we got 3 up here, we got 1, 2, 3, 4, we can 6, and we can do meiosis. And that's how polyploidy happens quite a bit in animals. In plants. I caught that. Those kind of errors, Heather, we try to correct, and Steve Cook has been instrumental in doing that for me in some of the edits, but I caught that one. Now, we'll return to polyploidy in a moment and its role in speciation, but now we're going to try to, and I want to point out that we're about to talk about pre- and post-zygotic reproductive isolating mechanisms. This is a post-zygotic reproductive reproductive isolating mechanism because it happens after the zygote was formed. And it's wasteful when that happens. The further you go down the path, the more wasteful bioenergetically it becomes. Now, that's okay. I'll let you read this. I'll read it for you. This first statement: variation patterns not only maintained but are in part established by reproductive isolation. If two different populations cannot interbreed, that means they can't exchange DNA. They tend not only to remain different but also to become further apart, increasingly different. In other words, that's how they diverge. If they can't interbreed, they can't exchange DNA, they're going to speciate or diverge. And there are two major ways that goes about: before the zygote is formed and after the zygote is formed. And as I said a moment ago, the second process that's actually manifested here is bioenergetically wasteful. And you try to understand what I mean is that if you stop the process, you reproductively isolate before you do bring the gametes together, it's less wasteful than if you do it after the gametes come together. All right, now the first one is the definition of a species by a zoologist. Ernst Mayr is probably the most famous one to come up with this. Species are groups of interbreeding natural populations that are reproductively isolated from other such groups. Now, that definition won't work for an asexually reproducing species, and there are a few that only reproduce asexually. And it's certainly somewhat of a joke. It certainly won't work. That species definition won't work for fossil species. I mean, banging two fossil rocks together is not going to do anything. This is sort of a joke, but so that definition is not foolproof, but it is it's called the biological species definition for a reason. And as long as if you're exchanging DNA between two populations, you're cutting down on it. You're cutting down on divergence. Now, some of the more obvious pre-zygotic reproductive isolating mechanisms of plants are the ones we're about to give examples of. And this is one of my favorite topics because it's so much where the rubber meets the road, so to speak. Now, I'm having real sinus trouble this morning, so excuse me. Now, events that lead to speciation of different races and species, starting with a homogeneously similar population cluster like we have on the left, what happens to it is you have at least two possible situations. You get a founder species becoming separate from the parent, and it's if you're trying to survive, self-pollination works real well in the founder population, but you eventually got to cross-pollinate to perpetuate yourself very long. But an allopatric population are those that are spatially separated. Okay, and so what we have here is an allopatric population, having been started from this second one being started from the founder species from the original population. And down here, we're looking at something that's called sympatric populations that have overlapping distributional ranges. And it's separated too from the original population. But what happened is during the separation, there was a development of reproducing isolating mechanisms more than spatial. So that when the overlap again, if that happens, there's a barrier that has developed. It could be mechanical or genetic incompatibility, but there's a barrier that keeps these two from interbreeding. So if you can have two species that are living together, without them crossing with one another, you know darn well that they're separate species. And so that's what's going on here. So I'll repeat that last one down here. At number 5, we have overlapping ranges spatially, geographically, but or even ecologically. But because of a reproductive barrier, they don't reconstitute into one species. They stay separate. It's easier for these to stay separate. In fact, the next slide is going to be separation like from Europe to the United States. The Western sycamore and the Eastern sycamore, the Occidentalis means Western and Orientalis means Eastern. So, but the hybrid between them is the London plane tree. Plantus with an X in their hybrid acerifolia, which is a hybrid of the two. So these two sycamores are geographically isolated. Now there's sycamores in the West that are somewhat geographically isolated from this, but Arizona sycamore, for example. But this is a geographical spatial separation. Sometimes it can be an ecological and physical sort of a microclimate difference. Oaks are notorious for being ecologically separated from one another, which is sometimes that in this case, we've got two look-alike, similar, at least in some ways, red oaks. And red oaks have bristle tips, whereas white oaks do not. But the red oaks are harder to tell apart usually from one another than is the white oaks, but usually. But scarlet oak is distinguished, is kept separate from black oak because they are ecologically have different habitats. The scarlet oak tends to like moist, poorly drained soils in wetland areas, wetter areas, whereas the black oak, which geographically, I'm down here and I don't have it, right? It's maybe 40, 50 miles for it. I go find some. It's not, we don't have the black oak in my county or my neck of the woods normally, but it likes dry situations. And if you bring them together though, they can hybridize. Now, another kind of, you're probably familiar with witch hazel. You're probably less familiar, and this is what's illustrated here. You're less familiar with the Ozark witch hazel, which does have the ranges do overlap, but they tend to bring on flowering at different times. Witch hazel flowers into winter or fall and winter, whereas vernal means spring, and guess what? That's when this other witch hazel comes to, that's when it flowers. So we've got a seasonal separation or isolation, which is what we're doing is going through these. And this is the timing of reproduction is the factor here, separating, keeping them separate. Milkweeds are notorious for having mechanical specialization or isolation that enables only certain pollinators to do the job. Orchids can have like a saddlebag configuration so that's what we mean by mechanical isolation. The specificity is so specialized, only certain pollinators can do the pollination. So that's an example of that. In fact, that makes milkweeds anatomically a very complicated flower to try to explain. You could spend an hour just sitting through and giving a class on the way these flowers have developed in terms of their structural complexity. And it all has to do with pollination and pollination, which precedes fertilization. Now, according to some estimates, 70% or more of the angiosperms are polyploid. That dummies, there's a book I talked about for them called Botany for Dummies. It's a dummies book, Heather. It says 50%, but so somewhere between 50 and 70% of the flowering plants are polyploid. That means they've got higher chromosome numbers than the regular diploid ancestors. Which that really means is that polyploidy has played an important part. And what I'm doing, if y'all have lost me, it goes way back. I'm saying that this kind of mechanism has gone a long way towards making angiosperm species what they are. Okay, now, if none of the prezygotic mechanisms that we've talked about work on keeping them separate or isolated, and if the hybrid zygotes are formed, there are some other mechanisms, and polyploidy is one of them, that actually keep them from, to keep the breeding population or species isolated, to prevent them from sharing DNA. And these are post-zygotic. So, and they're energetically, we said, bioenergetically more wasteful. Now, these mechanisms include any of the physiological or genetic process that would cause the hybrid zygotes to die if they survive, if they survive to become sterile. Let's see. In other words, polyploidy stabilizes the reproductive process and affords a degree of reproductive isolation that permits further evolution of the new species. Now, but if the hybrid survives, hybridization followed by back crossing to either the parent's plant leads to the integration of the new genetic material of one species into the gene pool of the other. Now, introgressive hybridization is a breakdown of genetic variability, and oaks are the best example. These are Western oaks, and this is what makes oaks so hard to identify, not just in the West, but here, is you get what's called introgressive hybridization. If there's a hybrid between something like a scarlet oak and black oak, like we talked about a moment ago, you get a hybrid. And then that hybrid crosses with one of the parents this way or crosses with one of the parents this way, then you've got what's the word? Well, continuity here. No distinct morphological separation, which makes these guys really hard to identify. And that's why when people can't identify oaks, they'll say it's a hybrid, and they're often correct when they say that. It's not just an excuse of inability to identify. So this is introgressive hybridization, and it's a way of muddying the water from the separation of two species. It establishes a genetic bridge, and that's exactly what's going on here, a morphological manifestation of that genetic bridge. This is pretty complicated. Have I lost anybody totally? Does that make sense? Okay, I can't tell because I've got the gallery view blocked out, but I hope. So now, let me make sure you understand here. The last bullet down here ultimately increases genetic variability or fitness of one or the other populations. If only one, then the other may die out and become replaced by the other. There are times when the offspring will become more fit in a given environment than either of the parents, and the parents could die out. But sometimes if all three survive, then you got three species. But this is a hybrid swarm. Introgressive hybridization is a fancy word for a hybrid swarm. Now, hybridization between two taxa will occur if the taxa are genetically similar enough. And this is a red wheat. Now, this number can be as low as 50, I've already said, according to some sources. In other words, most of the angiosperms are probably coming about by polyploids instead of, in the animal world, that's not the case. Now, here we've got two diploids, wheat species that are crossing one another, and you got a hybrid, and that's a sterile hybrid, and because there's no homologous pair of A genes and no homologous pair of B genes to do any meiosis, you got to have duplication. So what happens with this is that, just like I showed you back on that, go back and show you what's going on there is the same thing that we were showing is going on here. Okay, there. All right, because what happened there was you got, let me get back to the wheat. What happened back there was you get at this point, to get to this point here, you've got mitosis without cytokinesis. They call it here an error in cell division, and that's what the specificity, I'm giving you what the error is. You get mitotic division without cytoplasmic division and a reconstitution nucleus. These, the products, the double A's and the double B's, get put back into the same nucleus. So this tetraploid is now able to reproduce, and it can cross with another diploid that we haven't talked about before, another species, a third species. And actually I guess 4 species, because this dihybrid is given, this tetraploid is given another name, can reproduce with this one and form a hexaploid down here. So, and that's an example of polyploid derivatives speciating. And actually, if they survive and can cross, then they can persist if the environment favors them. Now, okay, what we're doing for orientation's sake is going back. If we go back to the slide from last week, we're going to talk now about propagule dispersal at the very end here, the last few minutes. And propagules have already been defined and redefined. And so we're going to go into that now. Okay, fruits and seeds. We're not going to talk about myospores, but all right. Wind, water, and explosive mechanisms can disperse the propagules. And in peanut, it can actually happen. What happens in peanut, I don't think I've got a separate slide for peanut for that, but what happens is that, I don't know if y'all realize it, but in the peanut field, when the flower doesn't last that long in a given day, in a matter of hours after flowering, the peduncle or flower stalk starts bending, and the developing fruit goes underground. Hypogea means underground. And so the peanut is not a fruit. It is a fruit. It's not a root. It's not a rhizome. It is a fruit that develops subterranean. And I don't know of any other, it must be, surely there's some other plants that do it. Among the 250,000 angiosperms, there's got to be some other thing that does it, the other plant that does it. But the flower gets pollinated, and then the fruit starts developing afterwards underground. And so it's a pretty neat way. And then something could even eat it down there and transfer it, but it's pretty unique, bearing its own fruit. And so now this is the wind pollination in willows is what we've already alluded to, and the poplars in the same family do the same thing. And I already said that willows have a mechanism that enables them to use some insect pollination because they have a gland. I don't know about poplars. I don't know if the poplar here has a little gland down here like the willows do. But they section this flower just to show you, actually section this catkin to show you what the arrangement of the different ovaries on it is. And then this is why cottonwood can be just like blowing around in the Midwest if you're out there at the right time. It's just, you can have just you have to sweep your foliar out because the poplar hairs are so common. And that's true of willow too. So not only do you have wind to pollinate the thing, you have wind we're talking about now to disperse the seeds, there's a seed covered with hair. So wind does not only fertilization, but it does dissemination of the seeds. Wind not only takes care of pollination, so fertilization can take place, but it takes, in the willow family, it also transposes the seeds. Now, this is a student of mine that got covered on his genes with the leguminous fruit. Now, most legumes don't have a pinching separation like that between the seeds, and they're called legumes. Whereas low mint is a special type of segmented legume that breaks up at maturity, and these little hairs on the fruit are kind of like Velcro. In fact, the guy that developed Velcro got the idea to do it from looking at plants. Burdock was one of the plants that he looked at in sunflower family, but these little uncinate, that's a term for hairs that look like little shepherd's hooks that catch on the clothing. And the degree of which they are there can help you to identify species of this tough to identify genus Desmodium. Tick trefoil is, I think, another common name for the genus. But you can see how that would, an animal with fur would do the same thing. Now, I've succeeded, I think, in getting rid of this from my yard when I was a kid. My sister and I used to throw these things at one another, throw a peduncle head on it, and it's to catch on your shirt. But coastal sandspur is one of the several species of Syncharis. And it can hurt if you step on it with a bare foot, I can tell you. That concludes our class, and right on time pretty much. And I hope that, let me stop the screen sharing. Okay, oh geez, all right, let me get rid of that, stop the sharing. And we're all done. And so are there any questions?
Bobby, I have a couple of questions.
Yes.
One is, can you give us a sense of how long it takes for two allopatric populations to speciate? In other words, you separate them, are we talking about millions of years, thousands of years?
It could be long times, Kevin, but not necessarily. It could be, all right, here's a case. There's a grass, a Spartina genus off the coast of England that its parents formed a hybrid. And that hybrid separated from the two parents within 20 or 30 years or less, I think. Certainly in the lifetime of a human, it could be that quick.
Oh, okay. That's
so
interesting.
In the plant world, it doesn't take long. It actually has to do really, Kevin, with the kind of thing we talked about earlier. Pine takes 2 years to complete a life cycle. Angiosperms complete it in a year. Some gymnosperms, like juniper, might complete in a year. But what you're talking about has to do with how many times you should shuffle the genetic deck.
Yes.
And if the longer growing season, a plant might reproduce more than one time in a year. But and in the case of a desert plant, the whole plant went from seed to seed in 14 days. So, shuffling genetic there, that of course, that's not going to happen in the desert, but probably once a year, or maybe on drought situations, irregularly numbers of years. So that's a good question. All right. Now, Kevin actually, I think, has a copy of this set that Donna had too, but not the final version, because I've added back slides that I cut out in order to do this in a short period of time.
Yeah.
But anyway, that's it. And you can give me your thoughts in an email if you want on whether or not we could informally do something like this. And I would send out an email saying on such and such a date, we're going to meet and talk about this topic if you want to join in. If you want to join in, let me know and I'll send you a Zoom invite. So I originally wasn't a fan of Zoom, but I'm becoming, after yesterday, I see the advantages in that meeting we had. It went very well. Especially for a long meeting of a board meeting. And I know.
Bobby?
Yes, Amy's got a question. Yes.
Yeah, I have a question. Back to the going from I guess the polyploidy. So you've got on that slide, you've got a sterile hybrid.
Originally, you get a sterile. Originally, you get a sterile hybrid because it can't, because something is odd number.
Right.
Can't, let me, before you get your question, let's get to that. Let's look at that here. Okay. All right. This thing here is sterile because an odd number, wait a minute. This A, it's like the A gene doesn't have a sister gene. And the B gene doesn't have a sister gene. And so once they fail to separate in mitosis and you get a reconstitution nucleus, the reason this individual here is not sterile, it's fertile, is because A has a homolog to pair up with in meiosis, and B has a homolog to pair up with in meiosis of B at metaphase 1 of meiosis. Now go ahead and ask a question.
Okay. I think we're talking about the same slide. So does that transform in one individual with some kind of a genetic error, or is this in a population?
Well, all it takes is one individual in a population, say 1 in 10, could actually do this. This little mistake, this little, oh, wait a minute, you can't see it because I'm no longer sharing the screen. I'm sorry. Let me, dummy of my part. Let me come back. Oh, geez. Okay, share the screen. And I'm not gonna do the annotate thing, but okay, now you can see that. All right. All right. Now, what I was trying, okay. But this homolog at this point, the reason it's sterile, there's no other A for this A to pair up with in metaphase 1 of meiosis. And this one here, I'm gonna have to, it's not letting me, okay. The B has no homolog to pair up with, and so they have to have this mistake. There has to be a mitotic division that makes 2 of these that don't separate into separate individuals, but wind up in the same nucleus. And so you, at this point, this triticum turgidum is no longer sterile because the A's have partners, the B's have partners, and they can divide. There's an even number. Up here, so that's what creates.
I understand. I think I understand that. It's just, does that happen often, or is it?
It happens in 50 to 80% of the time of species in angiosperms, they believe. So, in other words, a tremendous amount of the 250,000 angiosperms, 50 to 80%, arose by polyploid speciation.
Okay, so it's fairly common then
in the plant world.
For that to happen.
Plant world.
Right.
In the angiosperms.
Right.
Not
okay.
Successful hybrids in the animal world. When you look, you got a mule, and you don't get much. It just doesn't happen. So polyploid speciation in the animal world is not like it happens in the plant world. Before I leave the screen sharing, does anybody else have questions?
I have a question, Bobby, regarding the
yes, Steve, go ahead.
Variation in populations where you gave the example of the bitter herbs would do better after herbivory.
Yeah.
because they were
Oh,
bitter.
the bitter herbs do better in terms of survival.
Okay, and what I was just wondering, would the opposite of that be true with a plant that has sweeter fruit? Because animals would prefer to eat the sweeter fruit and then disperse the seeds elsewhere and make that plant survive.
Well, there are other species that don't operate with like they make the fruit so desirable. And of course, we're talking fruit, not leaves. Although
Right.
technically the covering on a fruit is a modified leaf, the carpel. But the bitterness doesn't get involved there in terms of selectivity. What happens is the fruit becomes so palatable or sweet or desirable that the animal prefers to eat it over the one that's less sweet, and therefore pass it through their system. And sometimes the passing through the stomach of an animal is necessary to break down certain inhibitors on the seed coat and make it germinate when it comes out the other end. So, and not only that, here's the seed. It's laying in a pile of poop, which is an organic fertilizer. And so the plant gets removed of its inhibitors to the seed coat, plus it's sitting there in a pile of poop, which is great for fertilization. So the plant has figured out without a brain that that's a good way to go. So, but it's all about natural selection. But that's an example of the opposite situation. And the sweet would have to do with the fruit as opposed to the bitter garbage it develops and stays in the leaves.
Okay.
And the reason the bitter garbage develops is because the plants don't have an excretory system to get rid of their garbage like animals do. Did I answer your question?
Yes, thank you.
Yeah. Any other questions? Okay, folks, it's been real. I think if I knew how much work it was, I'd never have done it. I'll probably never do it again. But the point is, I don't have to because I do have this recorded. This is recorded, and I don't know if I'll ever make it public or not. But right now, if you want to see any of it, as you well know, except for Heather may not really, you have to email me and I'll give you permission. And the best way to go about it is to have the slides. The real good way, I think, would be if you had this whole PowerPoint and then let me see here. It's not going to let me do it. Let's see. No, I can't change the views. Let's see. This is the normal view. I was looking for the notes view. Okay. All right. Yeah. The notes. It's not letting me show it, but the notes down here at the bottom of the slides are what you really need. To get to that, because that sometimes I'll actually read them, and most time I didn't. But this version is nowhere on any. I mean, the final version of 365 slides is not up for anywhere. No one has that, but I can make it available. In fact, what I would do is I think I've got one of these versions of earlier versions of the PowerPoint up on, I can't remember if it's Google Drive or the OneDrive of Microsoft. But what I would do is I would take down the old version and replace it with this version. So anyway, and that way y'all could download it with my permission. And now Timothy, speaking of downloading, Timothy Estep, which is not, he's a vice president of our class, has just on the Facebook part of our Georgia Plant site, which I don't look at. In fact, I don't do Facebook unless I have to. But there's some good stuff going on. And one of the things he did was make available or tell people the links to the biodiversity library. And you can download a 1,200 something page volume of the Manual of the Southeastern Flora by Small, which is the original book that I used to cut my teeth on when I was starting out in the 70s, late 60s, early 70s. And Small, it's the Manual of the Southeastern Flora, it's 1933. And Small was a splitter, and he split things out that people thought weren't good species, and now we're finding out that they were good species. But it is so big it crashed Chrome 2 or 3 times. And so what I did was I don't like Microsoft Edge that much. Well, I don't like it well enough to use it on a regular basis, but I use Microsoft Edge browser to download it successfully. Now, my next goal is to try to take that paid version of Adobe Acrobat that I have and try to make it text searchable, OCR recognition, which probably will crash Adobe, but I'm going to try it when I feel like it. But anyway, I have I can't search for the word Polypodium aviculare in the version I've downloaded, but if I do a OCR conversion successfully, I've never tried a document that big, I will be able to do that. So the beauty of the small illustrated the first species of the genus that he describes. And so that's sometimes when you can't identify something somewhere small will let you do it, but it takes a lot of, I know a person that had no botanical training has been able to use small successfully, so I know it's possible. Okay, people, I'm going to stop it there, and I appreciate.
Bobby, I just want to interrupt to tell you, I've seen a couple of comments in the chat section, but thank you so much for all the time you put into this.
Yes,
Yeah.
yes. Thank you, thank you.
Thank you, Bobby.
You're welcome. I'm going to try to look at Steve always gets after me because I failed to look at the chat thing, and I'll let's see if I can save it here. Let's see. She says, okay, let's see. Now let's save the chat. So I'm going to do that. And so that's been saved. But I appreciate everything and your patience and indulgence of me and my tangents sometimes. But y'all been the beauty of this, I think, has been you're not in a regular college or even high school class. You have to stop at certain points to cover testable material, and we didn't have to. We weren't locked into that, and that was good. The advantage is there's no teeth in it for you, but you just pure motivation on your part, and that's a wonderful thing, I guess. All right. So, all right, if you're interested in me doing something informally on a somewhat regular basis, let me know, and I'll try to save up 2 or 3 topics, not too many, don't want to go more than an hour because people have other lives. But anyway, so I appreciate everything, and there's only really been 6 or 7 of you. It's out of 400, and I just. It's a shame that probably 20 other people would have got out of the 400 might have profited from this. All of them would have profited, but what I'm saying is if folks knew what they missed, I think we'd have had more. So, but that's the way life is, right? All right, I'm going to end it.
I agree. Thank you very much.
Thank
All
you.
right.
Thank you,
Now-
Bobby.
Thank you.
Yes.
Thank you,
Thanks, Bobby, and thanks for letting me add.
All right.
Thank you.
Thank you.
Bye, Bobby. Thank you.
Bye, you all. Love you. Bye.